Morning, welcome. I'm Elizabeth Youngman. Is my mic working here? Thank you. I'm Elizabeth Youngman. I direct public health programs here at the Pew Charitable Trust. We're delighted to have everybody here with us today. I'm going to run you just quickly through what to expect today. After a brief welcome, Dr. Gottlieb will give his remarks, and then he's going to have a chance to do just 2 or 3 questions from the audience today. But then we have a fantastic panel of FDA leaders from across this policy area. And so we will have that opportunity to have a robust discussion of Dr. Gottlieb's remarks. We're delighted to see so many people here today, and just want to do a special shout out to those of you who are in our overflow rooms. You should have Pew staff in the room with you, so if you have any problems with audio throughout this event, please just flag your Pew staff person who's probably waving to you right now. Also, you will still have an opportunity to ask questions, so feel free to just flag down your Pew staff person, and they can get those questions up here to me and to the panel. So more from me later, but I am delighted that our Executive Vice President, Sue Uran, will be welcoming Dr. Gottlieb this morning. So let's get to the main event. Sue.
Thank you, Elizabeth, and good morning, everyone. I am Sue Uran. I'm the Executive Vice President and Chief Program Officer here at Pew, where we have a diverse mix of projects ranging from consumer health, state, and environmental policy to biomedical research and environmental research, and support for Pew's hometown of Philadelphia. It's my pleasure to be here today to welcome you and our special guest, Dr. Scott Gottlieb, the Commissioner of the FDA. Today's event addresses one of the greatest public health crises of our time, and that's antibiotic resistance bacteria. Antibiotics now cure diseases that once took millions of lives, but they also provide a crucial underpinning for much of modern medicine. Without effective antibiotics to prevent infections, procedures that we all take for granted today, hip replacements, chemotherapy, organ transplants would be too risky to undertake. The implications of a post-antibiotic world are almost unimaginable. And yet at a time when the need for new antibiotics has never been greater. Pew's analysis of antibiotic development pipeline shows that there are only 11 antibiotics currently in clinical trials that even have the potential to address the pathogens that have been designated as the most critical threats by the World Health Organization. Now, at the same time, the world continues to overuse the antibiotics that we have, accelerating the development of resistance. Now, Pew's research, in partnership with the CDC, shows that 1 out of every 3 antibiotic prescriptions that are given in doctors' offices and emergency rooms are completely unnecessary. Now, those are disheartening statistics. Nevertheless, important progress has been made. Since Pew started its antibiotics work in 2008, we've seen progress on many fronts on the battle against superbugs, with credit to many of you here in this room. Public-private partnerships such as CARB-X are helping to advance the development of promising antibiotics that are urgently needed. Hospitals are reporting more data on their antibiotic use and resistance patterns, and many food producers are rising to the challenge of reducing antibiotic use while protecting animal health. Of course, much more work remains to be done, and agencies across the federal government have recognized this and are taking action, including the FDA, which brings us to our guest. The people in this room are well aware of the extraordinary career of Dr. Gottlieb. He is a physician, a medical policy expert, and above all, a public servant. He understands the magnitude of this threat and is deeply committed to meeting the challenge of antibiotic resistance. His current position is Dr. Gottlieb's 3rd tour at FDA, so he has a deep understanding of the agency he leads and the important role that FDA can play in tackling this problem. So please join me in welcoming FDA Commissioner Scott Gottlieb. Commissioner Gottlieb.
They were probably worried I touched the top of the glass. Yeah, it's good we did that since we're talking about antimicrobial resistance here. Thanks for having me here today. I really appreciate it. It's a pleasure to be here. This morning we went live with a new website that consolidates the policy work going on at the agency around antimicrobial resistance, and I will tell you that it's probably been harder to pull together a multi-center website than to get the policy formulated. So it was quite a feat. So I hope you'll all have a chance to take a look at that. I want to thank Pew for hosting today's event. I know all of you are here today because you understand the importance of addressing this issue, the issue of antimicrobial resistance, and the need to maintain advanced tools for modern medicine, not only for this generation, but for many generations to come. The CDC estimates that in the United States alone, every year, at least 2 million people develop serious infections caused by antimicrobial resistant pathogens, things like MRSA and other bugs that I've grappled with myself as a hospitalist. And at least 23,000 people die as a direct result of these infections each year. We can't count on outracing drug resistance, but we can use stewardship and science to slow its pace and reduce its impact on human and animal health. To do so, we need an all-hands-on-deck approach to combating AMR in both human and veterinary settings. We need all of the above strategy to what we do here today. Today, I'm announcing FDA's 2019 strategic approach for combating AMR. This is an ongoing agency-wide initiative to build on our past efforts and to coordinate policy and external partnerships to help confront these challenges. This strategy has been developed by the agency and center leadership in FDA's Center for Devices and Radiological Health, our Center for Drug Evaluation and Research, our Center for Biologics Evaluation and Research, and our Center for Veterinary Medicine, and our National Center for Toxicological Research as well in Arkansas. The agency's Office of the Chief Scientist has also been instrumental in helping advance these initiatives. We're also introducing, as I mentioned at the top, that new webpage today, which will provide our stakeholders a one-stop shop for following the FDA's work on AMR. And we're launching in tandem with the release of the Center for Veterinary Medicine's 5-year plan. We're launching this in tandem with the release of the Center for Veterinary Medicine's 5-year plan for supporting antimicrobial stewardship in veterinary settings. So all of that is being unveiled here today. My remarks today are focused on FDA's role in combating AMR, but we don't undertake this work alone. We work very closely with government partners and also with many stakeholders, some of whom are here today and joining us by the webcast as well. And we're deeply appreciative of these governmental collaborations. The FDA, though, plays a unique role in advancing human and animal health. Our regulatory goals put us in a key position to help coordinate and advance these shared purposes. And so when we talk about promoting and protecting public health in the context of product development, we often talk about addressing the full continuum of product development and use. This comprehensive approach spans the entire life cycle of the product, from pre-market development to clinical trials to the need for post-market safety and surveillance. Across this entire continuum, the FDA works to ensure that products are safe and effective for their intended use. In addressing AMR in this context, the FDA is also adopting a comprehensive approach with 4 major components that address the full continuum of product development and use as well. And this includes steps to better facilitate product development and efforts to promote product stewardship, efforts to promote antimicrobial resistance surveillance and advanced regulatory science. We aim to address all aspects of this challenge. Because of the FDA's statutory responsibility for assuring safe and effective products that promote and protect both human and animal health, we have a unique vantage point for coordinating all of these aspects of a product's development and application to health. And different centers within the FDA have varied roles for assisting this comprehensive approach for safety and proper stewardship. CDER and CBER, for example, have a large role to play in helping to bring to market new antimicrobials and non-traditional alternatives to small molecules antimicrobial drugs. Most existing antibiotics are as old as the earth. They were literally screened out of the soil from nature where they resided, doing battle with bugs for centuries. So we need to find new ways to safely accelerate this evolution in our laboratories. CDRH also has a key impact on stewardship by helping to bring new diagnostics to the marketplace. These tools can help us rapidly test a patient to tailor treatment responsibly and effectively, targeting the right drug to the right bug at the right time. And CVM also plays a very critical role in these efforts in all portions of this product life cycle. But they have a very especially important and unique role to play within the FDA when it comes to helping advance better stewardship efforts in the veterinary setting. Our new comprehensive strategy is aimed at bringing all of our centers' work together via one platform to address 4 key areas. First, we're facilitating product development to ensure a robust pipeline of safe and effective treatments that can help combat resistant organisms. Second, we're promoting antimicrobial stewardship. Careful stewardship across human and animal health can help preserve the effectiveness of available treatments and may help slow the development of AMR. Third, we're supporting the development of tools for surveillance and antimicrobial use for determining when pathogens develop resistance. And finally, we're advancing scientific initiatives to help all stakeholders answer critical questions related to antimicrobial resistance. This includes research that can help support the development of alternative treatment approaches. These can include bacteriophages, fecal microbiota, transplants, and live biotherapeutic products. We don't have much time today to do these things, so I'm only going to touch on briefly on each of these 4 areas. I don't have a lot of time to cover all these in detail, but I know that the panel that's going to be speaking after me is going to get into a lot of the details of what we're announcing here today. First, I want to talk about facilitating product development. To sustain a strong antibiotic drug pipeline, we need a combination of push incentives to support research and development, as well as pull incentives to attract more investment. A lot of the focus so far has been placed on developing push incentives. I think much more emphasis needs to be placed on developing pull incentives. These can create natural markets for drugs targeted to rare but dangerous multi-drug resistant pathogens that we know can threaten human health. We've already seen some successes from both approaches. The GAIN Act is one example. It was passed in 2012 as part of the Food and Drug Administration's Safety and Innovation Act. GAIN was designed to stimulate the development and approval of new antimicrobial, antibacterial, and antifungal drugs. The Act created incentives for sponsors to bring antibacterial and antifungal drugs to market that are intended to treat serious or life-threatening infections. And it allows FDA to designate certain antimicrobial drugs as qualified infectious disease products. The FDA began granting these designations to qualifying products immediately following the enactment of the GAIN Act in 2012. And from July 9th, 2012 through August 2nd of 2018, where we have data, the FDA has designated 160 QIDPs. This includes about 83 novel drugs, and since enactment of GAIN, 15 QIDs have been approved by the FDA. The Limited Population Pathway for Antibacterial and Antifungal Drugs, or LPAD, was another piece of legislation created by Congress in 2016 under the 21st Century Cures Act. LPAD was created to advance the development and approval of antibacterial drugs to treat serious or life-threatening infections in limited populations of patients with unmet medical needs. 3 months ago, the FDA issued a draft guidance to assist in the development of drugs using this additional important pathway. I know there are many people in this room today who helped work on these important pieces of legislation. The draft guidance that we issued, when finalized, will support drug development by describing the criteria, processes, and other general considerations for drugs approved under the LPAD pathway. And we're encouraged that we're already seeing very meaningful early interest by innovators in potentially developing drugs under this new framework. The FDA expects that preclinical and clinical programs for drugs eligible for approval under this pathway will follow a very streamlined approach to clinical development. This includes potential for smaller, shorter, and potentially fewer clinical trials. But we continue to see a need for additional pull incentives in the form of reimbursement reforms that allow innovators to capture more of the long-run social and economic value generated from the development of new antibiotics. Large pharmaceutical companies have, for the most part, exited from antibiotic research. And while some small venture-backed companies remain engaged, these companies are not as well positioned generally to fund the larger confirmatory trials required for regulatory approval. What we'd like to see, most of all, is a balanced level of investment and more interest in these opportunities. We'd like to see a more robust market for these products, complemented by a more robust pipeline. We don't have that today. As large companies pare their efforts back, the funding pool available for commercializing innovative projects is shrinking. And we've recently seen some news that at least one big pharmaceutical company is getting out of this space entirely. So I'm deeply concerned by these trends. I'm concerned that without stronger pull incentives that encourage more research and development, we're going to continue to see far less robust pipeline of products than we need to address AMR. The problem is that the ideal drug is one that will be seldom used. Under the current reimbursement scheme for drugs, though, profits are driven off of the number of prescriptions that get written for a drug. But when it comes to developing medicine to treat multi-drug resistant microbes, the goal is to basically hold such a drug in reserve and try not to use it. In other words, the reimbursement scheme is in direct conflict to the public health goals in this context. And what we're seeing as a result, I think, is a classic externality problem. Creating new antibiotics isn't any easier than developing a drug in any other class, although it's sometimes been said that it is. It's a challenging pathway. And the commercial challenges in this context are more daunting. Generic antibiotics are also inexpensive and widely effective, making them first-line treatments. And Medicare's inpatient prospective payment system for hospital care is pegged to the price of generics. So novel drugs end up eating into the profits of hospitals when they're used in the hospital setting, and most drugs that are used for antimicrobial resistant pathogens are ultimately going to be used in an inpatient setting. And when new and better antibiotics are available, these novel drugs are reserved typically for last-line cases when their unique profiles and their higher prices are justified because first-line drugs have failed. It's good news because hospitals are engaging in better stewardship, but it also creates complexities to the model. It's completely justifiable behavior that we're doing this. If a new product is overused, it can reduce the effectiveness by increasing antimicrobial resistance. In-hospital stewardship has improved a lot. I know when I was practicing as a hospitalist not too long ago, several years ago, I had to get sign-off by the infectious disease team before I prescribed drugs. And this is all good news that we're taking better care of these drugs. But by holding a drug in reserve, it also shrinks product revenues early in the drug's patent life, when those revenues are most valuable to companies and investors. So the economics in this category and our public health prerogatives are not entirely in sync. Contrast this with markets for new antivirals for HIV and Hep C, where new medicines, when they've been introduced, they've replaced a number of older drugs becoming first-line treatments right out of the gate. And investment in these categories has followed these economic realities. If we want to maintain a robust pipeline for antibiotics, it's necessary to change the perception that the costs and risks of antibiotic innovation are too high relative to their expected gains without weakening antibiotic stewardship. It's important to pursue new policies and reimbursement approaches now to shift the investment landscape right away into these areas. And it's important to develop these products so that they'll be available when we need them later. One constructive way we could start to develop innovative payment mechanisms that allow companies to capture a greater upfront share of the social value of antibiotic drug development is looking at different approaches to reimbursement. The long-run human and economic cost of antimicrobial resistance is enormous, including death and disability from sepsis, extended and expensive hospital stays, and need for dialysis or organ transplant in the wake of systemic infections. The CDC estimates that the direct cost of antimicrobial resistance in the United States to the U.S. economy is on the order of $20 billion annually. When you factor in the economic consequences of lost productivity, it adds an additional $35 billion in costs. Reimbursement reforms could include a mix of milestone payments and subscription fees for developers of FDA-approved products with high economic and clinical value, targeted at multi-drug resistant organisms and linked to proven clinical outcomes. A subscription-based model in particular could see hospitals paying a flat fee, a flat rate, for access to a certain number of doses of an important new antimicrobial. These subscription fees could be priced at a level to create a sufficient return on the overall investment to develop drugs with a certain profile. This should have the effect of creating a natural market for drugs that meet certain important specifications. The FDA is actively discussing approaches like this and continued innovation in similar payment models with other agencies, including CMS, as well as private sector partners. New approaches to reimbursement that could be explored might also include new technology add-on payments for certain new antibacterial drugs that meet critical patient and public health needs. The Gates Foundation, BARDA, who's here today, Rick Bright, thank you for joining us, and the Center for Medicare and Medicaid Innovation could all help us join with commercial insurers to provide a natural testing ground for these kinds of innovative payment models. And these discussions are going on. These payment models might include the subscription-based model that I spoke about, and these incentives might be linked to the promise of effective stewardship efforts by providers and manufacturers to make sure that we maintain good oversight of the drugs that come through such a pathway. The FDA is also committed to facilitating innovation through proactive engagement with sponsors and researchers to identify and address challenges to the development and approval of novel and non-traditional antibiotic products. The FDA's Biologics Center is actively promoting development of non-traditional products in three major categories. These include bacteriophage, live biotherapeutic products, and fecal transplantation. Other relevant biological products include vaccines to prevent primary and secondary infections caused by AMR microbes. The best way to prevent a resistant microbe from becoming resistant is to prevent patients from getting an infection in the first place. So steps we can take to help cut down on infections in the hospitals really are the front line of these efforts. By reducing hospitalizations that put patients at increased risks of requiring AMR, vaccines can be a critical tool as well in reducing overall burden of AMR. Academic investigators, small and medium enterprises, and even large companies may have questions about the development pathway for their biological products. Some of those questions benefit from early discussion with CBER to facilitate product development. We saw this, for example, when it came to the development of fecal microbiota for transplantation. These discussions can help answer important questions, remove roadblocks, and ultimately help create a clearer route to getting safe, effective products to market and to patients. CBER's Interact meeting program was recently created for sponsors to engage with CBER staff and obtain advice on a specific topic or issue that's critical to early product development. The advice provided by CBER staff to a potential sponsor during Interact meetings may help streamline development by, for example, helping sponsors avoid unnecessary preclinical studies. And these are very early engagements that we're trying to promote through this new program. The FDA will continue to advance new ways to help facilitate development of non-traditional antimicrobials, as well as through a number of planned guidances that we'll be issuing over the coming months and year. The next phase of the product continuum, and another key part of our comprehensive approach, is implementing good antimicrobial stewardship. As all of you know, antimicrobial stewardship activities span the work of many federal agencies. We especially rely on the work of CDC in this area, and we work very closely with them in this regard. But we also have a responsibility for a critical component of stewardship in the veterinary setting here at FDA. These settings are an area where, as a nation, we've made important progress over the past few years in improving antimicrobial stewardship. Since I became commissioner 17 months ago, conversations with my colleagues in CVM have made clear to me that this progress is a result of close collaboration among a variety of stakeholders in this community. The FDA's two guidances for industry, GFI 209 and GFI 213, which I'm sure many of you are familiar with, combined with our updates to the Veterinary Feed Directive regulation, laid out a clear path for CVM to work with the industry to revise labels on all medically important antimicrobials used in feed and water of food-producing animals. The aim was to eliminate production uses of antibiotics, such as growth promotion, and the goal was to bring all remaining therapeutic uses under oversight of licensed veterinarians. The successful implementation of these changes depended heavily on the commitment of key partners and stakeholders to work with FDA. This included the animal pharmaceutical and feed industries, the animal agricultural community and veterinary community, as well as other federal and state agencies. And we're very pleased to see such a high level of collaboration and support to implement these changes. Ultimately, we were able to reach our goal of successfully updating 100% of the nearly 300 affected products. And because of this work, about 95% of the total quantity of medically important antimicrobials sold or distributed for use in food-producing animals are now under veterinary oversight. This is significant public health promise and progress. But our work, combined with the steps many of you in this room have taken to create your own stewardship initiatives and your own new product lines and new marketing campaigns, has together resulted in a very different landscape today in how antibiotics are used now versus how they were used for decades. But there's still a lot of work that we know needs to get done. And today we're announcing plans to address remaining areas where there can be further alignment with the principles of antimicrobial stewardship. One area of focus we're announcing today is our intention to work with the industry to bring the remaining 5% of medically important antimicrobials approved for use in animals, such as certain injectable antimicrobials, under veterinary oversight. Toward these goals, we'll release a draft strategy by the end of 2019, likely as a guidance document, to assist industry in making needed changes to their products. Another important area of focus is ensuring that medically important antimicrobials are labeled with appropriately defined durations of use. The FDA has determined that about 40% of approved medically important antimicrobial drugs used in feed and water of food-producing animals include at least one indication that doesn't have a defined duration of use. That's why FDA is announcing today our plans to develop and implement a strategy to address this issue. Now, implementing this change to all of these drugs is going to take time. So to advance these goals, we'll be releasing a draft strategy, likely as a new guidance document. We recognize that stakeholder feedback is going to be crucial in finding a workable solution. In the animal stewardship space, we're also committed to working with key stakeholders, both domestically and globally, to help users of antimicrobial products have the information they need to make these products available in a judicious manner. Toward these goals, we're renewing our commitment to work in consultation with our partners at USDA and to collaborate with stakeholders to identify ways to develop and disseminate information on antimicrobial stewardship. This ranges from collaborating with producer groups to provide information on stewardship to working with veterinary medical associations and academic institutions to create veterinary curricula with the most up-to-date information available. We're also taking new steps to support stewardship in human medicine. And to advance these goals, the FDA has worked with manufacturers on the development and availability of new generations of devices that can identify bacteria and fungal pathogens directly from specimens such as blood and spinal fluid, stool samples, sputum, without the need to wait for growth in cultures. In many cases, these tools can reduce the time for diagnosing the precise cause of a disease from days to hours. Several tests can also identify the presence of antimicrobial resistant genes, significantly enhancing the ability to select targeted treatments, reducing broad-spectrum antibiotic use, and improving patient outcomes. Lastly, FDA is working closely with manufacturers and academia on next generations of biomarkers to rapidly identify whether a patient's symptoms are due to a bacterial infection, or, for example, when antibiotics can be stopped, thereby avoiding substantial unnecessary antibiotic use. I want to move on to the third component of our comprehensive approach, gathering data on antimicrobial use and resistance. This is essential to understanding the drivers of resistance in the human and veterinary settings and formulating appropriate responses. But acquiring this data is complicated. It requires collaboration and coordination across multiple government agencies. The FDA is especially grateful to our colleagues at USDA and CDC who partner with us on many of these data collection efforts. We also appreciate Congress's interest in FDA surveillance efforts, and we're committed to making the most of the funds that have been appropriated to us and entrusted with us for these activities. One such activity is the National Antimicrobial Resistance Monitoring System, NARMS, co-managed by FDA, USDA, and CDC. Our veterinary center released a plan today that discusses further improvements we plan to make to pursue with respect to NARMS. For example, we'll be expanding NARMS sampling, supporting research in advanced genomic technologies and bioinformatics, building on a domestic capacity to monitor AMR and previously unexplored pathogens to include companion animals and animal feed. We're also working to further refine industry reporting of antimicrobial sales data, and we're seeking to gain a better understanding of how these products are actually used on farms. This includes continuing to fund several pilot projects to help us better understand on-farm use. We plan to publish our reports on this work. In human medicine, it's been long recognized that differences in hospital reporting on which specific AMR tests were conducted and how results of these tests have been reported has substantially hindered the ability to track antimicrobial resistance and implement decision support for treating physicians. Working in partnership with more than 40 other stakeholders, including CDC, the National Library of Medicine, pharmaceutical and device industries, and others, our Device Center's Office of In Vitro Diagnostics and Radiological Health has led the development of interoperable standards for the transmission of device codes from manufacturers to clinical laboratories. This work has supported the development of a microbiology coding manual. This new resource describes the effective implementation of about 12,000 microbiology codes into consistent electronic diagnostic results. Reducing inconsistency in reporting test results should substantially improve the reporting of antimicrobial resistance. Continued development of this program, called the Systemic Harmonization of Interoperability Enhancement for Lab Data, or SHIELD, it's pretty well named, should unlock the ability for real-time clinical support across all electronic health records. It will enable efficient tracking of prevalence and spread of AMR within and across institutions, and it will also advance the development of real-world evidence for different treatment approaches. Our Office of In Vitro Diagnostics and our Drug Center colleagues have also published a draft guidance titled Coordinated Development of Antimicrobial Drugs and Antimicrobial Susceptibility Test Devices. Not as well named. This policy can facilitate the availability of diagnostic devices to test organisms for antimicrobial susceptibility at the same time or very shortly after approval of a new antimicrobial drug. And we'll be working to finalize this guidance very soon. Coordinated product development between drug and device developers can be very effective in streamlining the regulatory pathway. Such coordination has resulted in antimicrobial susceptibility devices being available almost immediately or within weeks of recent new drug approvals. In addition, the FDA has received more than 13 pre-submissions from antimicrobial susceptibility testing, or AST, device developers. Recognizing that the availability of antimicrobial resistant bacterial strains has been a significant challenge to developers of diagnostics to test for antimicrobial resistance, the FDA and CDC collaborated to create the Antimicrobial Resistance Isolate Bank. This is a publicly available resource that provides panels of curated bacterial and fungal strains to device developers, to academic researchers and pharmaceutical companies, clinical laboratories, and many others. The AR Isolate Bank is an invaluable national resource, and to date has provided more than 2,000 panels for use in device development, in drug development research, and other laboratory uses. Each panel has been developed to focus on an important clinical concern. This bank has contributed to 13 clearances or approvals for AST devices, along with the development of other infectious disease test submissions since its launch in 2015. Advancing innovative AST devices and diagnostic tools and enhancing interoperability of IT systems will bring us much closer to real-time surveillance systems for antimicrobial resistance. This will enable clinicians and policymakers to monitor local and national trends, to design agile clinical trials, testing targeted treatment approaches without waiting many days or many months for reliable data. And finally, we're also pursuing our own intramural regulatory science initiatives. For instance, the FDA is developing methods to assess medical countermeasures and validating next-generation in vitro diagnostics platforms. In addition to these efforts, the FDA is also working with a variety of public and private partnerships to inform approaches to drug development, to facilitate guidance development and provide recommendations to streamline the efficient development of new antibiotics. To take just one example, the Biomarkers Consortium of the Foundation for the NIH has brought together industry, FDA, NIH, and academia to advance the science of clinical trial endpoint development for antimicrobial drugs. The Clinical Trials Transformation Initiative is another example. This program supporting research and expert meetings on streamlined development of drugs for serious bacterial infections with unmet medical needs. Through this effort, we're developing consensus-driven recommendations to streamline clinical trials for hospital-acquired and ventilator-associated pneumonia. And today, FDA is releasing a request for information to obtain additional external input on how we can best develop an annual list of regulatory science initiatives specific for antibacterial and antimicrobial products. This input will help the Office of Antimicrobial Products develop FDA's fiscal year 2019 regulatory science initiatives. And this will help FDA identify research areas where regulatory science can help support new antibacterial drug development. And this includes new efforts to create drug development tools or standards for use by industry or other stakeholders to better meet patient needs. There's much more work that we're doing at FDA to combat antimicrobial resistance that my colleagues will discuss shortly on the panel. I just scratched the surface on the things that we're doing and the things that we're announcing today. We're confronting, quite literally, a global public health threat. And we need to harness science and policy to help our public health systems and researchers become nimbler in the battle against drug-resistant pathogens. I'm confident that the efforts we're launching today and the ones that will continue in the months and years to come and the work that we'll do together will help us achieve our goals of facilitating stewardship and advancing innovation across human and animal health within our borders and across them. Thanks a lot. I have questions for,
I was gonna say, just as a reminder, we have time for just one or two questions. We're going to have a great panel afterwards.
sorry.
Go for it.
I could take a couple of questions. I hate doing this, but I know there's some press here, so I'd like to just prioritize them if I can and see if there's time left over. So if any of the reporters have any questions, and if not, fine. Ike?
Thanks. I'm Ike Swetlitz from STAT. I was curious if you have talked specifically with CMS about the specific subscription model that you're talking about and what sort of the time frame is for that turning into a demonstration or where that's going to go.
Yeah, it's still early days, and so we have had discussions with them, and we've had discussions. I've talked to SEMA, my counterpart there, and we've had some discussions with CMMI as well. Adam, I think it's still very early in trying to see what ability we might have to work with CMS in this regard. Could be something done through CMMI in the form of a demonstration. But I don't want to just focus on Medicare as a vehicle for trying to address this. I think this is something we might be able to do in concert with the private sector as well. Gates Foundation's expressed interest, others have. I think what you'd want to do, the subscription model wouldn't encompass the whole market. I think the goal would be to have a subscription-based model, for big institutions that are most likely to use drugs targeted to multi-drug resistant organisms that would provide sort of a foundation of return on investment, if you will. It probably wouldn't encompass the entire market for a new drug, but it would provide enough of a foundation of a guaranteed market that it would help more companies get over the hurdle rate, if you will, of making investments in new drug development in this area. And this is not, this is an idea that's been talked about. I've talked about it in the past. There was recently some discussion of it at a Duke event sponsored by Mark McClellan. But it's also a concept that we use to pay for a lot of other things in the in-hospital setting. Software is paid for this way. A lot of durable medical equipment is paid for on a subscription-based model. So hospitals are used to paying for things on a subscription-based model where they pay a flat fee for access to something, and they might pay a variable fee based on the number of beds they have or the number of admissions they have or for a certain number of uses. So these models are very well established. They've just never been applied to the drug setting. But as I said in my remarks, if the goal is to develop a drug that hopefully you don't have to use, that there's no market for that. And I think that we could work in concert with the private sector and maybe with other government agencies, including Medicare, to work on some kind of collaborative process here where we might have a role to play in sort of helping to define the parameters of what such a drug would look like, and if a drug meets those parameters, then the reimbursement mechanism would pay for it under a different approach that would be more of a subscription model. So we provide a very clear upfront incentive for trying to develop drugs that have these kinds of profiles. I don't know if you had any follow-up or if that answered your question.
I'm Sarah Carlin Smith. I'm with Politico. And talking about the payment models, is one of the things you didn't mention is possibility of government stockpiling. Would there be a role for the government to play in just paying upfront for certain antibiotics that we would need to have on reserve to encourage development?
Yeah, I think the issues around stockpiling are, considerations made in a sort of a different public health context. It's more of a national security decision whether or not something needs to be held in reserve. I'm talking about, decisions made in more of a clinical context, and so I think that they're separate. I don't think that stockpiling alone creates a sort of sustained natural market for a product that it's going to create the kinds of incentives for development that we want to see. I've been on the other side of this, as you know, and I've seen discussions in boardrooms and elsewhere around whether or not a certain product might meet the profile of something that would be stockpiled as part of a strategic decision, and that was rarely, if ever, in my travels, the pivot that made the decision to make an investment. It was a nice to have. It wasn't the point at which you made an investment decision, because what you wanted was, because that was always a binary decision. You weren't sure if the government was going to stockpile it. You weren't sure how much they would stockpile. You weren't sure how much that stockpile would turn over. So it was always highly variable, and the market was the market models were easier to make and to try to predict, and so that's where the decision got made. So we're thinking really in that context what we can do. I think the decisions around the strategic considerations are different.
All right. Well, thank you very much
Thanks a lot.
for taking the time to be with us. I think I speak for everybody in the room and just expressing our appreciation for your taking this issue. With that, could the panel join me up here as Dr. Gottlieb is leaving? Thank you. Somebody's mic's on. All right, well, with that, we'll move right to the second half of our program, a panel discussion featuring senior FDA agency officials from across the agency. It's really so exciting to have you all just here in one place at one time. I will briefly introduce our guests, and then we will have a lively discussion, I hope, about Dr. Gottlieb's remarks. I'll first ask some opening questions, and then we're going to open it up we should have some good time for audience questions, both from folks in the room and for those of you who are on WebEx or Twitter. And if you have a question, I just want to mention, if you have a question and you're not here in the room with us, either mention it to your Pew staff person, or you can tweet it to us at FDA@Pew. That's FDA@Pew. So with that, to my immediate left here is Dr. William Flynn, who is a veterinarian. He serves as the Deputy Director for Science Policy at FDA Center for Veterinary Medicine. Pew has had the pleasure of working with Dr. Flynn on these issues for a number of years, and we really appreciate you being here with us today.
Thank you.
Next to Dr. Flynn is Dr. Edward Cox. Dr. Cox is a physician who serves as the Director of the Office of Antimicrobial Products in the agency's Center for Drug Evaluation and Research. His office's work related to the development and review of antibiotics and antimicrobials is important for helping new products make it to the market. I'm sure we're going to talk a lot about that today. He's also able to talk to us about the identification of antimicrobial breakpoints, which for anyone who cares about stewardship, that's an issue near and dear to our hearts. Dr. Gitterman is also a physician. He serves as the Deputy Director of the Division of Microbiology Devices in FDA's Center for Devices and Radiological Health. Dr. Gitterman will help us to understand more about the intersection of antibiotics and medical devices, including diagnostics. And last but certainly not least, I'd like to welcome Dr. Carolyn Wilson. Dr. Wilson is a microbiologist who serves as the Associate Director for Research at FDA's Center for Biologics Evaluation and Research. CBER regulates complex microbiological products. and she'll be able to provide us further insight into some of the more cutting-edge science that will help us tackle the antibiotic-resistant problem, like using the microbiome to try to treat antibiotic-resistant infections. So welcome to all of the panelists. I'm going to start first with you, Dr. Flynn, because you're next to me, so in the hot seat. The commissioner outlined some plans related to kind of an ambitious agenda with a number of components to it. Could you help us get an understanding of what's happening when? When can we expect this plan to sort of officially be announced? What are the sort of milestones that we should be looking out for in the next 6 months to a year?
Sure. Well, certainly it was important that Dr. Gottlieb outlined sort of a broad strategy for FDA generally, which is, I think a significant step for us in terms of highlighting the sort of cross-cutting nature of the issue and the fact that, as he, I think, said, is we need a sort of all hands on deck kind of approach where we're looking at implementing activities, across all sectors. Certainly from the Center for Veterinary Medicine standpoint, we're focused on the veterinary component and the use of antimicrobials in veterinary settings. So the plan we are announcing today for related to CVM is focused on sort of identifying a number of activities that we think are important for helping to support and foster antimicrobial stewardship in veterinary settings generally. Our focus to date has been largely on the animal agricultural sector, but I think we need to look more broadly. So some of the elements in this plan also look towards the companion animal environment arena too. It is laid out as a 5-year plan, and I think the sort of focus for that these are the actions we're going to focus on over the course of 5 years. We've sort of divvied it up into sort of a phase 1, phase 2 in terms of mainly just trying to organize around, there's a lot of work to do and we can't sort of tackle it all at the same time. And so we're sort of phasing that in over that 5-year period. As far as timeline, I think the 5 years really is, the time, what we're focused on in terms of initiating over that 5-year period. I think the expectation is that we cannot, accomplish or complete every one of those action items. I think I counted 32 individual action items in our plan. We're not able to sort of complete every one of those, but I think our goal is that for each one of those, we're going to engage with affected stakeholders to flesh out more specific implementation plans for each of those different projects. I mean, some of them we have sort of in the sort of first phase already have some work done. The duration of use issue, which Dr. Gottlieb mentioned, we've already done some groundwork on that issue in terms of getting public comment. We are beginning to gather input in terms of developing a specific plan of how we're going to implement that, and also then as part of that plan, sort of set out a more specific target date for completing that. As Dr. Gottlieb mentioned, we're setting a goal for ourselves to get an initial plan in place to address that issue as a draft, where we will see public comment on that by the end of September of 2020. So that's one example, but there's many projects within the overall plan that we need to do more in terms of engaging with stakeholders to sort of map out what is sort of a practical timeline and plan for actually implementing all the measures that are in there.
So picking up on duration limits, that's something that FDA sought public comment on in 2016, and I know you announced that something was coming this summer. And so, what are you expecting the challenges to be as you move forward with that? How do you expect FDA to sort of move past those so that we can?
Yeah, I mean that's probably of the items that are in our plan. That's probably one of the more challenging of those 32 that I mentioned. And I think one of the what comes to mind with that question is the fact that I think the good news is that I think based on the steps we've already taken, all those products are now under veterinary oversight, I think, which was a critically important sort of first step to bring the veterinarian into the loop in terms of how those products are being used. But the fact of the matter is clearly these products, and so we're talking about therapeutic uses that are still, for which these products are being used. And there are some significant animal health issues or diseases that these drugs that farmers, ranchers, veterinarians have relied on or are relying on to effectively manage those diseases. So I think the challenge is, and I think what is that fact, is that we still need to be able to address those important diseases. They are sort of core to how, in some cases, how animals are being raised in the U.S. And so I think that's a challenge: is how do we sort of move past that? How do we address that? The concern we have about trying to better optimize these uses of these drugs, but at the same time make sure that we still have that tool available to help manage those diseases. So I think in terms of how we're approaching, I think one approach we're taking is that we're trying to focus on the driver. In other words, what is the underlying disease conditions that is sort of driving the need for using this product, and sort of go at it by looking at that disease condition and working with the industry, both the ag industry, the veterinary community, to figure out what are there better ways or alternative ways we can manage that disease condition so that we do not have to rely on these antibiotics as much as we currently are. So I think that's the approach. And again, the desire is we want to maintain the availability of these tools. We just want to make sure that we're optimizing their use.
So sticking with the labels issue for a minute, and then we'll get to the rest of the panel. I know that the missing duration limits aren't really the only problem on some of these labels, and there are a number of labels that otherwise depart from what FDA has described as being judicious use principles. So those with specified indications like weight maintenance in times of stress, those types of labels. Are your plans focused really on duration limits? Your label plans, at least, focused on duration limits, or have you considered sort of pushing into those other areas to ensure?
Yeah, I think there has been a lot of attention played on that specific issue. I think it's important to recognize that these products, this whole sort of inventory of products, which are also the products that we made changes to under the Guidance 209, 213 effort that Dr. Gottlieb mentioned. These are products that have been in the marketplace for many years, decades. And these products originally were approved as over-the-counter products. So the labels are written originally towards the layperson, because that's how they were originally approved and designed. Clearly, we've now made the shift that these products are under veterinary oversight. So, and so I think there is an opportunity to sort of look at these products through a different lens, which is these are now products that should now be in the hands of a veterinarian. I think so. That's an important piece that I think we are going to look at as part of this. So I think it's a little bit broader than just simply focusing on that duration question, but are there things we can do to sort of upgrade how these products are labeled, taking into consideration that the veterinarian is now a key part of sort of providing guidance in terms of how those drugs are being used, at the farm level.
Thank you for that. Dr. Cox, I imagine there are a lot of questions in the room about the market proposals that Dr. Gottlieb laid out today. I've heard you describe the pipeline as fragile, and certainly our pipeline analysis, which is available in the back, would suggest that. But can you help us what's your perspective on what is needed to reinvigorate this market?
Right. So, thinking about the development of an antibacterial drug, you start with the discovery of a new lead molecule. And that needs to enter into clinical development, and then if it's successful, into phase 3 trials, and then subsequently into the marketplace. So you can see if you're trying to have successful antibacterial drug development, you need to tend to all these stages along the pathway to development of a new drug. And if we look at what's gone on over the last decade or so, there are successes here, and I think it's important to recognize those. The folks at BARDA, NIH through CARB-X are paying attention and trying to facilitate the development of new lead molecules. So CARB-X serves a very important role in developing new lead molecules. NIH working in the area of early development, NIH along with BARDA, and then also BARDA picking up on later development to bring new drugs to the point of marketing applications coming in. So I think we've got some good successes there. And it seems that where we are now is that when a new product gets out there, a new product enters the market, the economic rewards are not to the point that they're able to sustain development in this area in the way that we need to in order to be able to meet patient needs. So that's really, I think where we are now when we look at antibacterial drug development and why, we are now looking more at the issue of, the economics of a marketed product and some of the discussion you heard today from our commissioner about potential, ways of looking at reimbursement that can help to deal with this last part that we need in order to be able to have successful and sustainable antibacterial drug development.
And what are your thoughts about how you not only bring and certainly we want to bring more antibiotics to market, but you also want to bring the right kinds of antibiotics to market. So how do you sort of target incentives to get the products you most want to have coming in the door?
Right. So my hope is that through CARB-X, what we'll see is a greater level of innovation. We've seen development. We're very grateful for the folks that are working in this area. It's challenging. It's not as economically rewarding as you would like, but my hope is that through CARB-X, what we will see are more innovative molecules that will be new classes that will have new mechanisms of action, and we there's also sort of a movement in the field, if you will, to try and look at some of these non-traditional therapies, so things that are different than what we're used to seeing from typical small molecule antibacterial drugs. So that's my hope, and the folks in CARB-X are working on this. We also recently did a workshop on non-traditional therapies to talk about some of the pathways. We went through a number of different examples of different types of drugs and how you would develop such things. The hope is to try and develop the science there and to try and deal with some of the uncertainty that might come along with developing a totally new type of molecule. So I'm hoping that we're putting in place the various pieces that need to be there to get to that higher level of innovation that we all hope to see that's really necessary to meet patient needs.
So this might actually then be the right place to skip out of order a little bit. And can you just maybe talk a little bit, Dr. Wilson, about the roles that non-traditional products could take or could play, in treating or preventing antibiotic resistance?
Certainly. Happy to do that. And Dr. Gottlieb mentioned 3 main categories of non-traditional products in his remarks. The first is a broad category of live biotherapeutic products. These are defined from a regulatory perspective as containing living organisms. They're typically bacteria. They may, in other settings, be referred to as probiotics, but when they're being used to treat, mitigate, or cure disease, they're now medical products, and they're under regulatory authorities in the Center for Biologics. The main mechanism of action here, or thought to be, is that these, what are thought to be therapeutic or beneficial, healthy bacterial populations may outcompete the populations that would be harmful and thus treat the disease that way. Another area that acts by a similar mechanism is fecal microbiota transplantation. And that's unfortunately exactly as it sounds. It's human fecal samples. And as you can imagine, there's a lot of promise and excitement around fecal transplantation. Check out DIY. I don't recommend that you do it yourself, but there's tens of thousands of hits if you do this. So there's a lot of interest in this area, but it's also very complex and challenging to regulate these products. Obviously, we want to make sure that they're going to be more beneficial than harmful. And again, the idea here is that they're going to be outcompeting the nasty bug, if you will. And then the third area is bacteriophage therapy, which actually has been around since the 20s or 30s. In the former Soviet Republic of Georgia, there have been uses of bacteriophage therapies, but not really in controlled clinical conditions. So a lot of our experience and understanding of these approaches are really more anecdotal. But these are viruses that infect bacteria, and the excitement here is that they are very precise. So unlike an antibiotic class that may wipe out a whole category of bacteria, we all know the impact on our gut when we take antibiotics. The idea here is, let's say you have a salmonella infection, you can treat with a bacteriophage that will just kill salmonella and not kill all the beneficial bacteria in your gut, which would be really exciting, right? But we need to make sure, again, that these are going to be safe and effective. And also, similar to drugs, there is the risk of developing resistance to bacteriophage, although the beauty of these are that there's an incredible plethora of genetic diversity of phage. And so the idea that some people are putting forward is that you might have a cocktail of phage that could be put together that targets a variety of different pathways, sort of like a chemotherapeutic cocktail, and therefore hopefully prevent development of resistance, or if resistance develops to one, that you still have other phage in that cocktail that could kill the bugs.
And the pathway to market for some of these products is a little more well understood, vaccines, antibodies, than for some of the newer products. And this may be a question for you. It may also be a question for Dr. Cox. But can you help? What is FDA doing? What can FDA be doing to help give guidance to folks who may be wanting to bring those types of products to market?
So we currently view that the current regulatory pathways provide sufficient flexibility to support licensure of these non-traditional therapeutics, as long as you can generate data that supports the safety, the efficacy, and the potency. Obviously, we're continuing to watch how these types of therapies are being developed, the data from clinical trials, and we'll reassess that. But we think that especially things like live biotherapeutics actually are very similar to live vaccines, which we have a lot of experience with. And generally, our center is very used to dealing with very novel and untraditional products. Our cell and gene therapies are always posing interesting challenges to how we regulate them. But at the end of the day, it always comes back to having the best available science and basing our regulatory decisions on that.
Thank you. So, Dr. Cox, do you have anything to add to that, or?
Yeah, so with development of non-traditional therapies, we do think there is a lot to be learned from development that's gone on so far. If you're studying a small molecule drug, or you're studying a non-traditional therapy for the treatment of a urinary tract infection, many of the ways that you would approach this would be the same. You'd be enrolling patients in a trial of underlying urinary tract infection, treating them for a period of time, looking at an outcome. So I think that there's probably more that we already know that we can utilize from development programs in general that will be applicable to non-traditional therapies. So that should help, because I think that tells us that there's maybe less uncertainty here than we would expect, because there are pathways that can be utilized that have been used for other types of drugs. So I think the challenges may not be quite as great as some might think for development of non-traditional therapies, at least with regards to the clinical trials.
So staying with drugs for just one more minute, Dr. Gitterman, we will get to you. But sort of flipping back then to traditional antibiotic products, arguably skin and soft tissue UTI infections are the easier indication. Not to suggest any of this is easy, but whereas perhaps what we need is have those types of indications. What would it take, do you think, to get companies to pursue those types of indications as first-line indications? And as part of that answer, I know a question we've got in the room, because I've gotten it a few times, is what role does LPAD play? And can you get an LPAD indication if the company is also getting a non-LPAD indication for that same product?
Right. So what we're seeing now is oftentimes, companies may initially pursue an indication in complicated urinary tract infections, complicated intra-abdominal infections, skin and soft tissue. So an indication where the time that it takes to complete the clinical trials, the cost, the level of difficulty is not at the highest level. And so the indications like HAP-VAP may be something that they're looking at to be a second indication. So if the drug can pursue a pathway where it can get to the point of a marketing approval, then from the standpoint of looking at a more difficult indication where the clinical trial might cost more, might take more time, as companies are making business decisions, they may be positioning this as a second indication, if you will. And the hope is that after that first indication, there will be interest in pursuing some of the more difficult indications. And it's not an unreasonable thing to do, because if you embark upon an initial trial and the drug fails because the test couldn't be completed, not because the drug doesn't work, then you've ended up not having a therapeutic. So if you can take this stepwise approach, that's certainly one way to try and get to the more difficult indications that are just simply more difficult to study. So that's one thing. The other is that we are working with folks like the Duke Clinical Trials Transformation Initiative to try and see if we can make the HAP-VAP trials easier to do. They are particularly challenging, but what are some of the changes we can introduce that can make it easier to enroll a patient into a trial, more efficient, and have a higher enrollment rate? We hear sometimes that you might have to screen 100 patients before you can enroll 1 or 2 into a clinical trial. So can we make it so that we better identify patients, look at the reasons why patients are not able to get into trials, and get a better rate of getting patients into trials to shorten the time of the trial and also to reduce the costs and to make it so that these more difficult indications can be studied. And then you also asked, what's the role of LPAD? So LPAD at this point is fairly early on, and we have a guidance document out there that tries to provide some clarity for some of the issues around LPAD. But we also recognize that there's still a lot of questions, and that's been reflected in some of the comments that we've received to our guidance document that's out there. And we certainly welcome the comments. They're very helpful to us. And as we think about LPAD, well, a couple of thoughts. One is that I think LPAD will become clearer after we have a few examples out there, because everybody will be able to look at those examples and see what the characteristics are of a drug that was approved under the LPAD pathway. So I think that'll be very helpful. That'll take a little bit more time, but I think that'll help to provide some of the clarity. And the other thing, too, is that when I think about LPAD, I think about a particular molecule that has certain characteristics, that has a development program of a certain type, where the results of the development program also are in sort of a certain category. So being an LPAD drug is not necessarily just something you wake up one day and you choose to do. There are certain characteristics of the drug. There's a certain characteristics of the development program. So these things all go together, I think, to get to what would be an LPAD drug, if you will. So it's something we look forward to. We look forward to moving the draft guidance to final, incorporating the information that we've received through the comments, and then also the experience that will be gained once we have a few LPAD approvals under our belt, thinking that will help provide some additional clarity on the pathway.
All right. And all of this drug discussion, of course, is more useful if you know what the patient has and you know what's going to work. So let's talk for a few minutes about diagnostics, which would be a game changer if we could have rapid point of care diagnostics that high sensitivity and specificity. Dr. Gottlieb talked a little bit about a number of data measures that are interesting, but what is it going to take to really get to that point?
That's a complicated question.
Can we do it in 30
This
seconds?
is what's no, but it is a complicated question in a number of ways. But we are getting there. The progress, and I would sort of second the theme that Dr. Cox, when you look back, of course we're not where we want to be. There's always progress to be made. There's a lot of progress to be made. But the progress we've seen over the last decade is nothing short of astounding in diagnostics. And I think there's several questions. One is, of course, the technology, but also, too, the science that's evolving is simply astounding. And it's actually coming together in concert. The new diagnostics we have out there are teaching us so much more. It's such a complicated question to answer it in a broad way. But I do think many of the things that Dr. Gottlieb had talked about actually are directly relevant. I don't want to say big data. That has different meanings to everyone. But the idea of accumulating data and sharing data and using it productively has tremendous possibilities. But also, too, as you've said, will diagnostics help with treatment? Absolutely. Just in a sense of point-of-care diagnostics that could separate a viral versus a bacterial infection, or non-infectious causes. How many people have seen poison ivy and they say, my God, what is that rash? Is it cellulitis? Diagnostics like that, by restricting, and I wouldn't say restricting, but by decreasing unnecessary antibiotic use, can make a tremendous difference. And exactly as you've said, for enrollment in clinical trials, if one could isolate quicker that someone has an infection, that what the sensitivity or susceptibility pattern of that organism is, that would make tremendous differences. I'd also mention that Dr. Dr. Cox mentioned HAP versus VAP. That's an incredibly difficult diagnosis in the present day. And diagnostics could help substantially, but data would help substantially. When we say biomarkers, we're talking about hundreds of potential biomarkers. The advances in basic science right now and the interconnectability of pathways, what comes before what intervenes. A simple issue like hydrocortisone. If there's any clinicians in the audience, the issue of when to give hydrocortisone in sepsis has existed since before I was born. Well, hydrocortisone was discovered not too much longer before I was born, but it's still there. But having said that, people are now isolating that there's certain biomarkers that can fairly readily distinguish who might respond to hydrocortisone or not. That would be incredibly effective for clinical trials. And so, have I answered your question partially? And I also do want to make one point about point-of-care diagnostics. And of course, that's tremendously exciting. And just a bit of caution, there are some diagnostics which unequivocally are point-of-care, home pregnancy tests, so to speak, where you, of course, and the risk-benefit for these high-performing diagnostics are important. Others may not be the best point-of-care diagnostics. Others may be far better done with clinical laboratorians who can do what we call reflex testing. One test generates the others. And, no test is perfect. And there's a lot of issues of what we call positive predictive value and negative predictive value that enter into decisions. So point of care is not a panacea. It's very important. And in some cases, it can be life-saving. Point of care arterial blood gases. But overall, it may not per se be a panacea. Just a note of caution.
Right, fair enough. Matching the diagnostic to the clinical decision that needs to be made, right? So maybe there's a broader way to talk about that. One frustration that we often hear from companies is about the delay between the approval of a drug and the availability of a related susceptibility test, in particular automated tests. Is there anything FDA is or can be doing to help kind of bring those timelines together?
Absolutely. And I think we've done certain things. And I would also say, too, anyone who has that we could do anything better, we are absolutely open. And we work very carefully with our partnerships with outside stakeholders, with industry and that. But I believe, as Dr. Gottlieb mentioned, there is a draft coordinated development guidance. And of course, there will be a final guidance, which actually will go even further, perhaps, than the draft guidance. But that has been, so far, very successful. Some of the fastest tests for doing susceptibility actually been cleared in CDRH. We use the word cleared, and somewhat analogous to approval, but not quite the same. And in the fastest review times that we've ever had, we're trying to put that model together. That time, ideally, will be eliminated. And I also think, too, another point that Dr. Gottlieb made, which I think has been invaluable, has been with our CDC colleagues, the National Antimicrobial Resistance Bank. I mean, this is something, and I have to give great credit to the people who led this, Dr. Ruby Schauer, Dr. Gene Patel at CDC. But one of the problems that was always seen is to test whether susceptibility assay is accurate, you have to have organisms that are resistant. And it's always been very difficult for companies. I mean, people say that, it's easy. It's not that easy. And FDA with CDC have got together these panels. Thousands of panels have already been. We had a manufacturer recently who was just effusive that having these panels to test and use during development expedites development tremendously. So we think, in quotes, again, predicting the future is difficult, but we think that this is going to go much more rapidly. We think we already have successes, but we anticipate even more successes. But we could always do better, no question.
All right. Well, thank you. That's at least promising.
It is, absolutely.
So we'll turn in just a moment to questions from the audience, but while you put your questions together, Dr. Gottlieb outlined an agenda, and I think we all want to be in a position in the fairly near term of declaring success, that we've made a lot of progress on each of these issues. You have got not just a room, but several rooms here full of stakeholders who are really invested in success in this area. And so just wanted to ask you, what can stakeholders, what can the policy community be doing to support FDA as you try to move forward? How can we help? Dr. Wilson?
I'll start. I think in our area of the non-traditional antimicrobials, the single most important thing that we're looking to the stakeholder community to do is to really support, develop, and implement appropriate controlled clinical trials so that we can, again, get back to having the best available data to support regulatory decision making.
Yeah, and I mean, the stakeholder community is a very broad group.
Oh yes.
So we recognize, no one person, no one group can solve this problem. I mean, everybody's got to work together. We need, the input from all the different groups that are involved in this space, and we see it in that we've had public workshops, and our public workshops engage folks from academia, folks from industry. We oftentimes have patient representatives there, and so that's a key part of our scientific work. But that's only one small piece, because there's so many other pieces that need to come together. We're grateful for all the other government agencies that are also involved in the work. So the stakeholder community, I think, it's really important that we continue to work together to solve the scientific problems, to address some of the other challenges outside of the scientific arena. We're talking about the issue of reimbursement, so that we can get to the point where we've got a sustainable enterprise that will be able to provide the drugs that we need both today and in the future.
Maybe just adding to that point is the fact that even just looking at the areas that Dr. Gottlieb outlined that we're focused on, is the fact that some of those areas FDA has more direct sort of control and influence and sort of, frankly, regulatory authority over, whereas other areas not as much. And so they're all important areas. And so whether it's looking at stewardship and what can we do to help with stewardship and foster stewardship, just looking from the perspective of the veterinary side, CVM doesn't regulate the practice of veterinary medicine, which is sort of core to implementing good stewardship. So for us to sort of make advances in that arena or that sector in terms of stewardship, really it's inherent that we have other stakeholders and others that are more directly involved and can influence that to be engaged in that. And so I think we're just saying, well, we're just trying to identify what can we contribute, but clearly to make progress, we need others too, because there's only limits to what FDA can actually do in any given sort of component.
Yeah, I would second what everyone else is, or I guess fourth what everyone else would say, that we have had, and again, I don't want to name drop, but we've had great success with some stakeholder communities. And of course, I would absolutely include manufacturers in that, but we've been able to test new models with the stakeholder community, which I think are so promising models, because part of AMR, of course, is responding. It's a evolving issue, it's responding very quickly to new pathogens. And we've been able to establish models where you don't have to come back to FDA, depending on what the governance structure, and there's groups out there. Of course, molecular diagnostics are so rapid. Every patient with an infection can contribute to certain to databases to some extent. And TB especially has been leading in worldwide databases where patients are being entered all the time as part of routine medical care. And with just a slightly more investment, slightly larger investment, these patients can contribute to relationships between new molecular mechanisms and clinical outcomes. It's very, very exciting. There's, again, to second what Ed had said, we're very interested in the science of new trial designs, which is certainly evolving in diagnostics. Studies like the RADICAL study, which the ARLG, again, I will name drop, very, very NIH-funded consortium. Many efforts, but Ed used the word, which I think is critical, which is sustainable. having sustainable clinical trial networks, and people can do this on an ongoing basis to make sure research continues, is very critical. And again, some of the stakeholders have been very, very helpful in putting down the models for these types of activities.
So a lot of focus here on clinical trials and evidence generation. So the panel has generously left another few minutes for questions. And so my colleagues, Andrea and Daniel in the back, have the microphone. So if you would like one, please signal to them. Here.
Thank you. Kim Walker from ASM. I've got a question that the commissioner sort of touched on but didn't elaborate. How do you see antimicrobial resistance in terms of national security?
You want to try? So, if we think about antimicrobial resistance, in essence, what resistance does is that it takes the therapies that we rely upon may lose their effectiveness. So that can happen really in any setting, whether that be in the patient in the hospital. If you don't have an effective therapeutic because of the development of resistance, then you won't be able to effectively treat a patient's infection. And you can see that concern could also be the setting. It could also have implications for countries and economies that could get to the issue of national security. And then also, too, if we're starting to think about biothreat agents, then we're also concerned about the issue of resistance because we want to have effective therapeutics. So that's just sort of a broad overview of the topic in general, and I hope that addresses your question.
Thanks. My name's Kate Bond. I'm from USP, and just building a little bit on that question with respect to global health security, we know a lot of these pathogens don't respect borders, and what we're seeing outside the U.S., given that we have a very secure supply chain in the U.S., is the problem of poor quality, substandard, or falsified antimicrobials. And there's a recent JAMA report indicating that 12.4% of antibiotics in lower middle income countries and 19.1% of antimalarials are estimated to be substandard or falsified. There's another WHO bulletin paper out recently calling on the need to look at veterinary products as well in terms of quality. That's a bit of a black box. I've just returned from the International Conference of Drug Regulatory Authorities last week, and there was a panel there of regulators discussing their roles from other perspectives in AMR and the importance of quality monitoring and surveillance. And I wonder if you could expand a little bit on some of your global cooperation efforts, both with respect to the human and veterinary products and surveillance of quality in particular. Thank you.
Right. So the issue of drug quality, adulterated products has been out there, and it's a particular concern in the area of antimicrobial resistance. Because if you're not receiving the antimicrobial or not getting the correct dose, that can certainly create a condition where there's a greater likelihood of development of resistance. So how do you address this? Well, it's a difficult problem, as you can imagine, because these are events that are happening in faraway countries that may not have the capacities to be able to do all that they would like to do in regulatory arenas to identify such projects, to prevent such practices. But there is work going on out there to try and improve the situation. There are efforts, both the FDA and also from private enterprises, private foundations, the Gates Foundation, to try and strengthen, and other private foundations too, to try and strengthen regulatory authorities around the globe. So the hope is, and it's not an easy problem to solve, but with time, with effort, that there will be strengthening of regulatory authorities to try and prevent such problems where patients are not getting the drug that they expect to get within a tablet or not getting the dose that they expect to get. Recognizing the particular importance of that. It's important in all areas, but particular importance with in the setting of using antimicrobial drugs.
And if I might bring Dr. Flynn in here too, because there's an analogous issue with agricultural practices.
Yeah, so I think some of the same comments apply. I think we are also engaged in the veterinary side in the international arena and with a variety of different organizations, and certainly put a fair amount of effort and resources into supporting capacity building and as a way of trying to get at the fact that there are certain parts of the world, certain countries where the regulatory infrastructure is not so great, and then therefore you have the possibility for products entering the marketplace and which are low quality. So yeah, I think it's sort of similar issues both on the human and the veterinary side.
Hi, I'm Mei Wu with the Natural Resources Defense Council, and I'm wondering on the animal antibiotic side, is given that when antibiotics are used routinely in animal agriculture, it can both exacerbate resistance and its efficacy, any efficacy it may have had in preventing diseases. So I'm wondering why CVM isn't focused on getting rid of that loophole and instead focusing on how to find ways of preventing diseases that don't require the use of antibiotics.
Well, I think if I understand your question, I think what we're focused on is looking at, obviously we've already taken steps to eliminate growth promotion or production uses. The idea behind that was the fact that from a judicious use standpoint in supporting good stewardship is that we should obviously try as best we can to limit the use of these drugs only when they're really necessary. So that is sort of a core principle that we make sure that we're only using these medically important antibiotics when there's actually an actual animal health issue that's being addressed. I think from our perspective, that may be through treating a disease or controlling a disease or preventing a disease. Having said that, then we need to make sure that those uses are as judicious as possible and that we optimize how those drugs are being used to manage those diseases. So I think as we talked about on this duration of use issue earlier, is that, and some of those products, some of those duration of use issues do sort of intersect with the disease prevention issue, that we need to look at all of those, what those drivers are. In other words, what are the drivers of antimicrobial use in different veterinary settings, and really look critically at those uses to determine, are there steps we can take to sort of better optimize how those drugs are being used to manage those diseases. So I think that, in theory, is what we are focused on doing.
You have time for one more if there is another one.
Hi, Brian Harvey, retired FDA. First of all, I just want to say, thank you so much for being here, because it really sends a very positive message to have the leadership of all the different centers coming and supporting this. And I do want to say, though, don't forget to educate your troops. There are frontline reviewers that still haven't bought in, and I don't mean to pick on CBER, but there is an example in the vaccine space. There hasn't been a lot of discussion, but to have a vaccine for Staphylococcus, even resistance, would be incredible. And yet, when you go and talk to certain reviewers, they go, "Well, what's the model? You need to study it in hip replacement, or you need to study it in knee replacement, and you just can't go for the broader market." The example from a few years ago, meningococcal B, which is a devastating disease that often results in death. There was a lot of difficulty for the two companies to get their product approved because you would hear things like number needed to treat versus the harm. And it wasn't until there was an outbreak at Princeton that it got the attention. So, educate your troops. You're setting a wonderful example, but sometimes the frontline reviewers have trouble thinking beyond, what's the model, what's the trial, how many trials do you have to do in order for those models, and because some still say you get what you study. So you've got a big job now, you've got the vision, and now it's the implementation that's going to be key, which ultimately will benefit the patients. So thank you.
Thank you for that feedback. It's helpful. I do want to mention that actually getting a vaccine for Staph aureus, since you did mention that, is quite complex. There's still a lot of gaps in our basic understanding of the pathogenesis and the approaches of how to develop a protective immune response that are really limiting the ability to get to an effective and safe vaccine. So it may seem sometimes like it's a first-line reviewer problem, but there is a larger problem in terms of needing to fill some of our important gaps in the science.
Well, that's unfortunately all of the time that we have today. So I thank the panel for being here. I also want to thank the Pew staff who put this together. It's been a lot of work. Melissa Rosenblatt and Kyle Kenner and many others in this room put a lot of work into putting this together. So thank you. We really appreciate FDA's role in tackling this problem. I think it's historic to have you all here together, and so very exciting for us. And thank you all for being here with us. Our pleasure, our pleasure. There are materials in the back. If you want those on the way out, please check out Pew's website for even more. And let's give this panel some well-deserved round of applause.